worship you with all of my heart, and I will worship you with all of my mind, and I will worship you with all of my strength, for you are my Lord. You are my Lord.
Dear participants, greetings from JMJ College Forum and Tenali. The Department of Chemistry is happy to welcome you all for the second day of the international webinar on recent innovations in pharmaceutical and applied sciences. Now I request Mrs. P. Hemalata Madam, HOD of Home Science, to introduce today's eminent resource person, Professor Ratnakaram Venkatnath.
Good morning, all of you. I feel it my privilege to introduce Professor Ratnakaram Venkatnath as a resource. It is a challenging task for me to concise the enormous accomplishments of Professor Venkatnath. Sir is working as a professor in the Department of Chemistry, Deputy Director for Admissions, and Research Coordinator in School of Science, Geetham deemed to be University, Bangalore, Karnataka State. He did his MSc and PhD in Chemistry, PG Diploma and MPhil in Biotechnology. He published 92 research articles in various national and international journals, and 5 more were at the doorsteps of publication. Most of these journals were Scopus indexed. He is author of 4 books and 9 book chapters. 4 research scholars completed MPhil, and 12 were awarded PhD under his able guidance. He worked in various positions in Vignan University as well as in Geetham University, where he is currently working. Receiving funds approximately 2 crores for various projects in a decade by different funding agencies is noteworthy and exemplary for his scientific outlook, hard work, and talent. He also guided 4 SSP projects. As a student, he is recipient of many merit scholarships and university rank holder at UG and PG. As a teacher, he received Best Teacher Awards in the year 2006 and 2010 by Vignan University and in 2018 by Geetham University. He is BOS member for various universities and editorial board member for International Journal of Pharmaceutics and Drugs Analysis. He is editorial member, reviewer, and board member for some other international journals also. He delivered more than 30 invited lectures on different topics in the field of chemistry at various colleges and universities. He participated in 9 FDPs, 14 workshops, 5 certificate programs offered through Coursera, 4 short-term programs, and 5 webinars. He is is the designer and organizer for 7 workshops and conferences. He presented 48 papers in conferences. He is a life member in 5 professional bodies, Associate Fellow in Andhra Pradesh Academy of Science, and Fellow of Indian Chemical Society. He instituted a gold medal for a topper in MSc Analytical Chemistry and Inorganic Chemistry, Acharya Nagarjuna University. If I continue to place his records before you, it takes another 2 hours. So I will stop with this. Sir, we are very fortunate to hear from you. Now I am handing over the session to you to proceed. Thank you, one and all.
Okay, thank you, Nirmala Jyoti Madam, for inviting me, and thank you, Parimala Jyoti Madam, for your good introduction. Thank you very much. I'm so thankful to the organizers and JMJ College for giving me an opportunity to interact with all the members. And due to some issue of this sharing, I have shared the PowerPoint presentation to Nirmala Madam and the organizers. If they are able to share it, I will continue my lecture. Madam, please share the.
Okay, sir.
Please. Actually, the sharing option is active, but whenever I'm trying to sharing, it is becoming inactive.
Okay, sir, no problem.
Yeah. Okay. Hope that my voice is audible. Madam, can I continue with this audio issue? No problem, na? No issue with the audio, right?
It is visible, sir.
Okay. So, audio
Audible also, sir.
Yeah. Thank you. Yeah, good morning to one and all. Today I'm going to discuss a little bit about chemical, biochemical, and microbial advances in process industries. Next, please. Next. Yeah, so when we take any industry, we are aware that there it is having many wings. If you start from production to quality assurance, quality control, and if you take the R&D, the bulk drugs formulation, packing, regulatory, sales, and marketing. So out of all these, the role of the chemist starts from production to quality control. That is the 4 main wings, whatever are there. The chemist is going to take an active role. Next one, please. So if you look at any process industry, what are the patterns and the innovations in the process industry? So when we look at the innovations, already we are aware that there are 2 types of innovations are there. One is the product innovation, the second one is the process innovation. Already you are aware the difference between these two, because I am not going to discuss much about it because of the IPR. So in India, more and more the industries go for the process innovation to get rid of those copyright issues and whatever IPR issues are there. Okay, the frequency of major innovation, you can find how they are going to related or correlated with each other. Next one, please. And then if you look at the pharma industry, it has been playing a vital role in the GDP of India. So if you can see here the comparison of the contribution of this pharma industry in the GDP of different countries like Brazil, China, India, and Russia. And from this, it is expected that even up to 2050, it is projected that minimum 5% of contribution will be there in the GDP of India with respect to this pharma industry. That itself shows the importance of the pharma industry in the Indian economy. Next one, please. So this is a comparison, and if you look at this one, how the exports and domestic wise, how many US $1 billion contribution is there. If you look at from 2020 to 2020, there is a very good increase is there. Of course, the percentage wise, there is some spike and some downs are there. So if you look at around 2004, you can observe a very good spike in the FDA inflow in the application of the product patterns. That is known to all of us. And similarly, sometimes you can observe that there is a slowdown is there. For example, if you look at around 2020, there is a global slowdown is there due to the US subprime crisis. So likewise, due to the various factors, both spike and slowdown is there, but on average, the Indian pharma industry is able to maintain a very good growth. Next one, please. And then if you look at this one shows the contribution of different products. So you can observe that the current size in 2020, the US billion dollars wise, it is around 42, and by 2030 it is expected to go or increase to $130. And this growth we can predict in terms of the business as usual or biosimilars or novel products. Novel products means known to all of us. Biosimilars means nothing but the biological medical product where almost identical copy of the original product, and that is manufactured by different manufacturer. That one is there. And in addition to that, adult vaccines and then biosimilars, adult vaccines, how they are going to contribute, we are able to see here. So both in terms of the domestic, where the yellow one represents the domestic one, whereas the gray one represents the exports. So it is expected that will be almost 300% improvement in terms of the US billion dollars from 2020 to 2030. Next one, please. So this is the contribution, how the chemical industry is going to rapidly increase. Okay, in addition to the contribution of the simple chemistry, other factors also influence that. One of the parameter is nothing but the digitalization. So how the digitalization is going to transform the chemical industry rapidly so that the value of the entire industry is going to grow. So let's look at understandable. This slide is a little bit what is called not visible much. Please, can you go to the next one? Yeah. So if you look at here, how digitalization is going to help this chemical industry or pharma industry means, so you look at here the first one, management of the feedstocks. So already you are aware that how this digitalization is going to transform the entire society. So based on this digitalization only, recently even the government of India has introduced some of the specialization even at the level of what is called graduation level, that too especially the BTech level. So nowadays you are aware that in addition to the regular BTech program, they are offering what is called minors, different minors. One is IoT, Internet of Things. The next one is AIML, that is artificial intelligence and machine learning, cyber security, data sciences, and chain management. These are the different digitalization initiatives taken by the government of India, and these are going to play a vital role. So how this IoT, AML, cyber security, data science, how they are going to help a chemist or chemical industry? Now let's discuss a little bit, not in detail. So if you look at this manufacture of the feedstock supply, so in this case, that one is going to help us. If you look at what are the feeds, okay, what are the raw material, how much is available, what is the cost, etc., etc. So the entire information, whatever is there, and that can be obtained with the help of the data science. So whatever the availability of all those can be known with the help of this one. Whereas if you look at R&D, R&D or the laboratory, so mainly in this case, the machine learning and the artificial intelligence are going to play a vital role, both in the research as well as the development. And similarly, if you look at here, the cognitive plants, in this case also, the PAT is going to play a vital role. So what is this PAT? How it is going to influence? Let's discuss in detail in the next slides. Okay, that is and then similarly, if you look at the last part, that is the management of the process, supply chain in the circular of the economy. Here also the digitalization is going to play a vital role. So being a chemist, we are not much interested in all these aspects. Let's look at where this digitalization is going to help for our chemists. Let's discuss. Okay, next slide, please. Now, okay, whatever I have shown earlier, okay, here there are so many factors are there which are going to help us to improve the process industry. What are those means? You can observe that the green part, whatever is there is a digitalization. The next one is how to utilize what is called the fuels, mainly hydrogen fuels like that. and electrification, and then the usage of the carbon monoxide and carbon dioxide, and then resource flexibility, and then symbiosis between industry and urban. So these are the different parameters. Out of all those parameters, let me discuss or concentrate on the digitalization. So when we look at the digitalization, already we have seen from the previous slides how the digitalization is going to help us to improve the process or product, and how the digitalization is going to improve us the plant and plant operations, and the digitalization how it is going to help us in the case of the value chain. So out of all these, in the case of the digitalization, we are going to discuss in detail about DOE approach only. Okay, next one, please. Yeah, so already we have discussed here. When we look at the digitalization of the design phase and the process and materials, in this case, there will be in silico modeling. Already you are aware that the in silico modeling, no need to discuss about that one. At present, what is happening when we take the pharmaceutical industries, the pharmaceutical phases are there: phase 1, phase 2, phase 3, phase 4. So whenever any product is developed, so to cross all these 4 phases, it requires minimum some 10 to 14 years. So even after 10 to 14 years, if the last phase, if it is unable to cross, whatever the efforts, whatever the money, whatever the time, whatever are there, entire will be a fertile one. So for that sake, whether a particular synthesized molecule is going to cross the 4 phases of clinical trials, that can be predicted with the help of this in silico modeling. Of course, not only for this clinical trials, nowadays we are using this in silico modeling even for the purpose of the synthesis. And then this is about the first one. The next one is the digitalization of the plants. So when we take this digitalization of the plants, okay, what happens whenever we are the production is happening? Naturally, the chemist is going to collect the whatever the reactant mixture from the reactor, and then they do the analysis in their lab, and then they give the information to the operating manager in that particular reaction plant. But what happens? Sampling, analysis, and then passing over the information, all this takes a little time. By the time probably whatever the unrequired results might be there in the plant. So in such cases, instead of going for the traditional sampling and analysis, nowadays advanced sensors are there. These advanced sensors, whatever are there, they are going to help us in the terms of the process analytical technology, the PAT, that is a process analytical technology. What is this process analytical technology means? No need to carry the sample to the laboratory. So within the reactor itself, there will be arrangement of the sensors. Those sensors are going to help us to learn about the conditions of the whatever the reaction conditions. Based on that, we can do the changes or manipulations can be done immediately. So in this way, the real-time optimization is going to be helpful whenever the digitalization of the plants is there. And the next one is the digitalization of the concerned process and supply chains. So in this case, of course, this one, the supply chains, whatever are there, it may go under this business analytics, etc. We are not much interested. We are interested in the first 2. Can you go to the next one, please? Yeah. So when we take this digitalization, how the computer, taking the help of the computer is going to help any process industry or a chemist. So we can see here. So when we take here the PCS, the process control system, PCS means the process control system, and the lab data, whatever is there, the performance, MES, that is nothing but manufacture execution system. So the process control system, this manufacture execution system, these are going to help us to give the data. And whenever the data availability is there, we can go for the accessibility and then connectivity. And then the data we must be able to store in a proper way. And then, okay, so where the data will be stored means already we are aware that nowadays instead of a server, the data will be maintained in the cloud, nothing but the web-based data storage is going to happen. And the thing is here, whenever the data is available, the main thing is about the security of the data. So probably you have seen that recently, whenever the COVID happened, whatever the Indian Bombay-based, whatever the industries are there, most of those industries where cyber attacks happened from other countries. So data security is also very, very important. And not only the data storage, data availability, the analysis of the data, which comes under data analytics. So in this data analytics, the visualization, and then proper modeling, and then process optimization, and predictive maintenance, and then performance prediction, all these come under the category of this data analytics. So ultimately, the big data success is happening when you start from this process control system to this manufacturer education system, data availability, storage in a proper way, and then analytics, then only it is going to help us for the big data success. Next one, please. Now, how these pharmaceutical companies are going to use this data analytics so that they can drive the innovation as well as the value. So when we take this one, the data analytics, we can say that mainly 6 parameters are there. Those 6 parameters we are going to discuss. So the first one, if you go for the discovery of a drug or the development of the drug, the acceleration of it can happen with the help of this data analytics. So that is very, very important. And then if you look at the efficacy and clinical trials, just now I told that whenever we take this what's called clinical trials, phase 1 to phase 4. So this one in silico studies are going to help us a lot. So optimization and improvement of the efficacy of the clinical trials is also possible with the help of the data analytics. And not only that, okay, at the level of the laboratory, a chemist is able to do the discovery and development was done, and then clinical trials also gone very well. But the problem or the issue is with the patient population. So whether we are able to target the specific patient population is or not, that is also possible with the help of the data analytics. And then, okay, not only that, even if you take this target specific patients and whatever the drug is there, whether the delivery is going to happen in a proper way or not. So improvisation of the drug delivery and effectiveness and the healthcare outcomes, that is also requires the data analytics. And then, okay, after the drug delivery, what is the result? Okay, whether it is safe or not, is there any risk is there or not? So that data analysis has to be done so that the improvement of the safety and the risk management, in this case also data analytics is going to play a vital role. And finally, as we said that this is very important for any industry with respect to the owner, that is insights into the market, the sales performance. In this case also data analytics is going to play a vital role. So from all these, what we can say, well, whenever we take the process industry, that too especially the pharma industry, the data analytics is going to play a vital role. So like earlier, a chemist cannot say simply, I know only organic chemistry, I can go for only synthesis, or I am an analytical chemist, I do the analysis of the lab, analysis of the drug. That is not enough. You must have the minimum knowledge of the what is called updation of this software. Then only you are going to sustain your life in the pharma industry. Next one, please. Yeah, so, okay, unlike earlier, nowadays there is a converging of the technologies is there, if you take the medical field. So not only the biological sciences, even the material sciences, the information technology, whatever we discussed here, the cognitive science, nanotechnology, they are going to play a vital role here. So next one, please. Now, okay, till the time I have discussed a little bit about the digitalization or the usage of the software and how this software technology is going to play a vital role in the pharmaceutical industry. Okay, to deviate a little bit, let me discuss a little bit about the one of the advanced technology, or not the advanced technology, but used technology more and more, that is the ultrasonics. Let me discuss a little bit about it. Then I will go how this DOE approach can be taken in the case of the chemistry laboratory. So I want to give you a little bit break to this usage of the software. Okay, let's go to the discussion of this ultrasonics. So if you look at this one, okay, this is very long back, 1894. The torpedo boats were tested. Okay, if you look at left hand side, actually this is the shiny new Francis turbine. But whenever the testing was done, after some time, the turbines were destroyed. The question arises why there is a destruction of the turbines. They came to know that that is due to the bubbles or the cavities from the spinning propellers. Whatever the propellers are there during their spinning, there will be formation of the bubbles or cavities. Those cavities are responsible for the destroying of these turbines. So what are these means? That cavities or bubbles are created because of this ultrasonics. Actually, for this discovery and extended work, the Lord Rayleigh got the Nobel Prize in the year 1904. Next one, please. So here, what is the so specialty of this ultrasonics? So when we take this ultrasonics, you can see here three-dimensionally. So one dimension is nothing but the pressure. The second dimension is the time, and the third dimension is about the energy. So if you look at this one, here it is showing the comparison of different types of the energies. So starting from geoenergy, piezo, thermo, flame, photo, sono, like that. So if you look at all these different types of the energies, if you look at the last one, whatever the required for us, the sonochemistry, nothing but the ultrasonics, this one, if you look at on the time axis, this one requires very less time. So within the less time, we can use this ultrasonics. So with respect means we can consume, we can save the time. So within the less time, it can happen. And if you look at this axis, energy axis, so energy wise compared to thermal, that whatever is there, ultrasonics is there, energy is high. So less time, energy is more, and the pressure, whatever is there, the pressures, you can compare this one. Okay, so with respect to either time or energy or the pressure, we can say that these ultrasonics are going to help us to complete the required reaction with less time by providing more energy and maintenance of the proper pressure. So when we look at the sound, generally we are going to divide into two categories. One is infrasonics. The second one is called ultrasonics. So at present we are interested in the second one. There is the ultrasonics where it is having the frequency more than 20 kilohertz. Next one please. So earlier, we know that even when we were studying the school level, the Doppler effect. So with the help of this Doppler effect, this submarines used to work. We studied that one. Following the same technique, whatever is there, Doppler flow meter is used nowadays to measure the speed of the red blood cells in our system. So the same technique, whatever is there, the same Doppler effect, whatever is there. Similarly, the CUSA is nothing but the Cavitron ultrasonic surgical aspirator. This is neurosurgeons are going to use this one. So whenever any brain tumor is present, that is effectively can be removed efficiently with the help of this CUSA technique. These are the well-known techniques in the case of the medical industry. Of course, now we are going to use it for our case. Next one, please. So you know that how these ultrasonics are going to help us with respect to the energy transfer. So look at here at the bottom, you can see that if you look at the y-axis, the bubble radius, and the time on the x-axis, the time is there. So if you look at from the beginning to the end, the initial stages, wherever the formation, the bubble is there, the bubble size or the radius is very small, and slowly with an increase of the time, so the time is very, very less. It won't require much time. That will be microseconds only. Within the microseconds, slowly there will be growth of the bubble. Whatever the bubble is there, after reaching a particular size, it is going to undergo implosion. So we should not mistake it. It is not explosion. It is implosion. Why not the explosion? Why the implosion is happening means, as the bubble size is going to increase within the bubble, naturally the pressure, whatever is there, it is going to be different and it causes the implosion. So just like in the case of the explosion, even in the case of the implosion, there will be creation of the shock waves. Whenever the shock waves are created, they are going to create a lot of high temperature that will be to the level of thousands of Kelvin. So what is happening? So there will be formation of a hot spot. What is the meaning of the hot spot? When we take ordinary general thermal system, in the case of the general thermal system, the temperature uniformity can be observed throughout the medium. So, but in this case, wherever the temperature is required, so the bubble implosion takes place and then hot spot is created, thousands of Kelvin temperature is maintained. So that is the main advantage. Means no need to create the uniform temperature throughout the conditions. Wherever we require the required temperature, at that place only this hot spot will be formed. And one more advantage is rapid quenching. What is this rapid quenching means? Wherever there is a formation of the hot spot, the energy will be transferred and immediately there will be a sudden decrease in the temperature is going to happen, so that the nearby system is not going to be affected much. So in this way, from the formation to the growth to the implosion, so the bubble size is going to increase. After the implosion, there will be creation of the shock waves, which are responsible for the maintenance of the hot spot, where the temperature is going to be transferred at high temperature to the successive stage, successive place, and then rapid quenching happens. In this way, ultrasonics is going to help us for any chemist. Next one, please. Now, if you look at this one, the black one, whatever is there, the black one shows nothing but what is called bulk liquid. The bulk liquid means the entire liquid, whatever is there, we call that one as the bulk liquid. The bulk liquid shown with black color, this is maintained at the ambient temperature or nothing but the room temperature. So whenever we look at the red circle shows the supercritical fluid where the temperature is maintained around some 1900 Kelvin. But look at here, in between at the center, there is a cavity of the gas where 5000 kelvins are going to be used. So in this way, whenever the imploding of the cavity is there, that is going to maintain the very high temperature, that one is going to help us to take place the reaction within the less time. That is the main advantage. Next one. Yeah, so this is the as we said that this one ultrasonics is used not only to carry out the reaction. Of course, nowadays these ultrasonic machines are used even in the jewelry shops. So whenever anyone who goes to jewelry shop and they give their jewelry, okay, and they say that they are dirty, can you please make it clean? So they are giving within 10 minutes or 15 minutes. So how means they are using this ultrasonic baths. So in this ultrasonic baths, what happens? There will be cleaning action takes place. But here, whatever the ultrasonic bath we use in the case of the extraction or the reaction will be a little bit different. So you can observe here, this is a sample, and then there is an ultrasonic probe is present, and then there will be a PVC screw cap will be placed on the vessel, and then the entire vessel is placed in the water bath where the uniform temperature maintenance can be happened. And then based on the requirement, the ultrasonics are going to be generated with the help of this 20k kilohertz ultrasound generator, and then the whatever the so generated ultrasonics with the help of the probe, it is going to help us to carry out the entire system. So this is the one which can be used for both the purpose of the extraction as well as for our chemical reactions also. So if you look at the bio laboratories, whenever the cell wall breakage is required, the cell wall breakage, they do many techniques, either using enzymes or the mechanical, or it may be ultrasonic. So just like in the case of that, even here the same type probe you can use here. Next one, please. So these are the ultrasonic reactors. And then if you look at the bottom right side, this is the one. This is ultrasonic bath, which we can see in most of our laboratories we use nowadays. We are using for the dissolving of any chemical. So instead of simple stirring, we are using this ultrasonic reactor. Similarly, the same is also used in most of the what is called shops, jewelry shops for cleaning. And here at the middle, you can find that there is a probe where you can use this probe insertion. That one helps us to take the reaction. These are the different supplies of this ultrasonic reactor. Next one, please. Yeah, so these are some of the papers. I'm not going to discuss about the reaction mechanism because already you are aware that except a few times the reaction pathway will be similar to that of the general case. And this is the work done by one of my scholars, sir, and he was able to prepare synthesize the novel 1,2,3 benzotriazole derivatives, where he prepared with the help of the ultrasonics without the solvent-free conditions. Already you are aware that nowadays the solvent conditions are going to be a problem to the environment. To maintain the proper ecosystem, naturally we have to reduce the solvent. That's why whenever we take the green chemistry, out of the 12 principles, one principle says that better to go or adapt the solvent free condition. Such a condition was adapted by the student, and then he prepared, and the antifungal activities were studied by him. Next one, please. This is one more paper where the synthesized compounds with the help of the ultrasonics in the solvent-free conditions, in vitro anthelmic activities were analyzed. Next one, please. Yeah. So with this a little bit introduction to the ultrasonics usage in the pharma industry, okay, how they can be synthesized within the less time and giving the more energy. Now let's go to the a little bit biosys side, that is stem cells. Nowadays, no need to give much information to anyone about these stem cells. These stem cells are very well known to all of us. And then umbilical cord, whatever is there, from that, okay, they are going to collect. They are going to collect these stem cells. They will be preserved. So whenever there is any health issue is there for that same baby, whenever they grow up, or their parents also, they can use same stem cells. So in this way, the stem cells market is there a lot nowadays. Now I am going to focus a little bit about these stem cells. So when we take this stem cells, all the products are very well known to you. The stem cells are converted into the adult stem cells, means the required organs can be prepared, or human embryonic stem cells are there. And then these stem cells, different services are nowadays available. So the number one, simple storage in terms of the stem cell banking, and then acquisition, testing, and in the case of the drug delivery, and then target identification, this one is going to help us a lot. And then when we take these stem cell technologies, the main and important one is nothing but in addition to the stem cell acquisition, the production, and the main and very high costly one and important one is nothing but the cryopreservation. So this, whenever from the umbilical cord, whatever the stem cells are collected, they will be preserved at a very, very low temperature, and they are utilized based on the requirement. And okay, so when we take this stem cell application, how they are going to help us? Just now I told you that in the case of the regenerative medicine, so whenever you are having any neurological disorders, the reason for that, then the replacement can be done so that they can be reduced. And then in the case of the orthopedics, so nowadays they are injecting these stem cells at their knee. మోకాళ్ళ దగ్గర ఈ స్టెమ్ సెల్స్ ని వాళ్ళు ఇంజెక్ట్ చేస్తారు. ఆర్థోపెడిక్స్ దే ఆర్ యూజింగ్ దిస్ వన్. అండ్ దెన్ ఇన్ ద కేస్ ఆఫ్ ద క్యాన్సర్, so what happens in the case of the cancer, there will be unnecessary required growth of the cells will be there. So whenever those cells are going to be burned with the help of the laser, so sometimes even the healthy tissues are also going to be damaged. In such cases, the regeneration of the required cells is can happen using these stem cells. And similarly, hematological disorders, the name itself indicates that some of the members who are having the defects with respect to the blood cancer, etc. In such case also we are going to use this one. And then cardiovascular diseases are there, what is called in such case we use. And then whenever injuries are there, in such case also we can use. Likewise, you can list out a number of the stem cell applications nowadays in the case of the medical field. Next one, please. Yeah, so when we take the stem cells, a number of the companies at present are there in the market who are doing an excellent work, like RLS, that is a Reliance Life Sciences, and then Life Cell, and then Cryobanks India, and then Stempeutics, These are the different industries which are doing a lot of research on it, and then they're helping to the humankind with their activity. So when we look at this one, okay, where the research is going on means, as I said, that these stem cells are going to help us in the case of the regenerative medicine, where a range of novel products are there, where the cell therapeutics can be done, where the tissue engineering can be done. Wherever the tissue engineering is there, in such cases, such related products, they require the stem cells so that they can get the engineered products. So likewise, we can give a number of the applications. I'm not much interested in that. Next one. Yeah, of course, in spite of so many applications, these stem cells at present, they are facing some challenges regarding the legislation. Yes, though earlier some problem is there, nowadays strict legislation is there, and rules and regulations are framing, and of course enforcement is a little bit difficult, but it is happening. And the issue is about the quality and ethical issues. So quality, okay, just like in the case of the chemical systems, okay, the quality maintenance is not so easy. Okay, quality control is going to be a different problem, and ethical issues. Okay, whatever the stem cells of a particular family, if they are going to be donated to others, okay, how are you going to accept it? That is also going to be one of the ethical issue. And the lack of awareness. Lack of awareness means, okay, all over the India, all over the world, including India, some stem cell banking systems are available. So those who wants to store those stem cells, they can do it, and in future they can reutilize it. But awareness is not much is there in the public or society. And then nascent success history. Of course, though we are saying that the stem cells are going to help us in a number of health sectors. Okay, the proven record is not at is there. Still, it is at the nascent level. That is one of the main challenge. Next one, please. Yeah, so when we take this design principles of these stem cells, the process components, the process function, and the process requirement, these are going to play a vital role in the case of the bioprocessing of the stem cells. Next one, please. Yeah, so the thing is here, when we take the stem cells, undoubtedly they are going to help us, but the problem is we are able to do at the laboratory level. So already those who are having some pharma background, they are aware that how the production is going to be. When we look at this pharma industry, there are different levels are there, different scales are there. So one is already you are aware that lab scales, whatever we do at the level of grams, whatever is there, that is the laboratory bench scale. So when we take this, though there is a lot of success at the bench scale, whenever you shift to the pilot scale, so you face many problems. So you know that the pilot scale means at the bench scales, whenever you go for the grams, whenever you go for some kgs like that, the pilot scale, it is going to be a problem. Already you know that what is a pilot scale? Pilot scale is nothing but 1/10 of the manufacturing reactor level. So that is called, in general, we call that one as the pilot scale. So at the pilot scale, naturally there will be some issues will be there, mainly with respect to the stirring and maintenance of the temperature and the collection of the sample. All these are going to be a problem. So from once if you are able to maintain the proper conditions at the pilot scales, naturally we shift to the next level and the last level. The last level is nothing but the production level. So in the from bench to pilot, pilot to the production level, whatever are there, the chemists are going to face some problem. In a similar way, just like a chemist, even in the case of these stem cells, undoubtedly we are able to grow at the laboratory level, but when we go to the large scale production, it is going to be a problem. So large scale industrialization is going to be one of the problem here. You may ask the one question. So what is the need to go for the large scale production of the stem cells? Because the stem cells are going to help us the series of the clinical trials, whatever we do, and then the drug development activities also require all these. Okay. So here there are different steps are there. One is the cell culture at the laboratory, and then scale up, and then characterization of it, just like our chemically produced compounds, and then proper enrichment, purification, and process control. All these are the different stages involved in the future progress of this large scale production of stem cells. Next one, please. So as already I have discussed this, so when we go from bench scale to large scale, in between the pilot scale, we face some problem. So all these are going to we have to do proper way. Next one, please. Yeah. So okay, when we take this large scale production, okay, how it happens? Already you are aware that when the case of the pharmaceutical industry, we use the reactors. And with a small modification, whenever we use, in the case of the microorganisms, we call them as bioreactors. Even in the case of the stem cells also, we can use such a reactors. Of course, there will be variation in the design as well as maintenance of the conditions from chemical reactor to bioreactor to and other reactors, the stem cell reactor. Definitely there will be some changes. But before understanding this, whatever the reactors we use for the stem cells, let us have a little bit information about the bioreactors which are used for the bioproducts formation using the microorganism. That helps us a little bit. Next one, please. Yeah, please look at here. So if you look at this is one of the fermenter. So when we take the fermenter or this bioreactor, whatever is there, as I said that the main conditions, whatever are we maintain, the design will be similar to that of the chemical system, but a little bit different. So how it is going to be? So if you look at the top, you can find the motor. The motor is having a shaft. Hello. All right. So here there is a motor. Motor is having a shaft and which is connected to the impellers, and then these impellers are going to help us to stir the medium. So what is the use of the stirring of the medium is to maintain the homogeneous conditions, so that what is called the nutrients availability will be uniform to the cells or the microbes, whatever are there. Of course, whenever we take these impellers activity, action will be good whenever there is a presence of some baffles. You know, baffles means nothing but at the edges there will be some baffles. Those baffles are going to help us to maintain the stirring conditions. So in addition to that, you can observe that there is a jacket. There is a jacket which is called cold water jacket. This cold water or cooling jacket, because you know, in the case of the microbial systems, the biological systems are mostly exothermic in nature. They simply release the temperature. So to maintain the proper maintenance of the temperature, we require what's called cooling conditions. The cooling water circulation has to be done. So at the sites, there is a presence of the what is called a cooling water circulation system, so that the temperature can be maintained. And then one more thing is here, the oxygen concentration probe. What is the oxygen concentration probe means? Okay, just like in the case of water, even in the case of the medium, there will be presence of the DO, that is nothing but the dissolved oxygen. So whenever we take the cells growth, definitely they require, in addition to the nutrients, they require DO, that is the dissolved oxygen levels. They are going to be maintained. So what is the level of the oxygen? So proper maintenance of the oxygen is possible if there is presence of a sensor which is able to which is going to help us to learn the oxygen concentration. So this is the oxygen concentration probe. And then if, for example, further oxygen concentration is required, in such case, there is a sparger. You look at the bottom, there is a sparger. And here a compressor, here the compressor air is filtered and then passed through the sparger. And the sparger means it is nothing but a perforated system. From the perforated system, the air or oxygen will be bubbled to the top, and the oxygen maintenance can be happened here. And then, so whenever we take this particular medium, always it must be free from the microorganisms. That's why we call this medium must be sterile nutrient medium. So whatever the medium we prepare, initially it is not sterile in condition. So that's why we have to make it sterile. How it is possible means, look at the top left hand side, there is a nutrient or the inoculant. Sometimes the same inlet is used even for the inoculants. Inoculant means nothing but the microorganisms or cells, whatever you say, we call that one as an inoculant. So the nutrients inlet is here. So after sending the nutrients inside, we pass the steam. What is the use of the steam? Means the steam is going to help us to sterilize the conditions inside. And then whenever the growth of the microorganisms or the cells is going to happen, there is the formation of the foam. So foam means automatically what happens? The headspace maintenance will be difficult. So you know that what is the meaning of the headspace means? The surface of the medium, to that of the surface of the medium to that of the vessel top, the gap whatever is there, which we call them as headspace. The headspace will be filled with the foam, and hence to avoid that sometimes the spillover also may take place. To avoid that, we are going to add the antifoams, and then to maintain the proper pH conditions. Here there is a need for the inlet of either buffers or acids, and then we have to take the measures so that there will be no much increase in the pressure. So to measure that, there is a pressure gauge, and then what happens whenever whatever the feed, whatever the nutrient you have given at the time of the conversion to the product, definitely there will be release of the some waste gases. Those waste gases are removed from time to time with a proper filtering system. This is the fermented system. A similar system, whatever the fermented system I have shown, the bioreactor I have shown, this is meant for the microorganisms. Of course, you have seen that some of them are having the similarity with that of the chemical reactor, and a little bit improvisation is there with regard to the biological system. There is a bioreactor. So further improvised system will be there, which we call a reactor, which we are going to use in the case of this stem cell production. Next one, please. Yeah, so look at here. As I said that, okay, this is a bioreactor, and you are giving the inoculum to maintain the conditions, either buffer, acid, or base you are going to give, and the substrate. The substrate is nothing but the nutrient. Inoculum means the cells, whatever you are adding. And this is a bioreactor. As I said that there will be continuous passes of the air or oxygen or carbon dioxide based on the conditions we pass here. And then we said that earlier we used to carry a little bit amount of the medium from here, and then we do the test. Nowadays it is not going to happen. In situ analysis. In situ analysis means NIR, near IR spectroscopy probe is there. This probe is going to help us to learn about the composition and the performance of this one. And then here, whenever off gas release is there, so before release of those off gases, always better to go for the analysis. That's why it is connected to a mass spectra. So everything is an online system. So this is a bioreactor process analytical technology, where you are going to do everything online. No, okay, so that a proper maintenance can happen. Next one, please. Yeah, so when we take this advanced process, we have seen what is called PTC. In this PTC, the advanced process control is required. How the control can be done? So for that sake, there must be a series of activities here. The process kinetics have to be followed from time to time. From this process kinetics, whatever the data you get, you have to analyze it properly, and then based on that, the process modeling, and then we will be able to give advanced process control systems, and totally we will be able to optimize the process. So in this way, from conventional control methods to there is a paradigm shift to the advanced control methods is possible. Next one, please. Yeah, the same thing with the little bit online system. Okay, time is not enough. Let me next. Yeah, so this stem cells growth to production, whatever is there, I won't to touch much on it because I will go for the DOE, design of experiment system, which is required for the chemist. I'll go a little bit more. So this is about the laboratory and the pilot scales, how they are going to be cultured. Go a little bit faster. Next. So when we take the biological bioreactors, already we have seen what is that, what are the different conditions we are going to maintain. Next. And then what are the advantage of the bioreactor means, as we said that unlike the laboratory level, proper scale up is possible, and the process monitoring can be done, and the reproducibility is also possible with this. And then enhanced mass transfer as well as the oxygen supply can be ensured in this case, and then minimization of the accumulation of the toxic or metabolic waste can be reduced, or minimization can be done. And then likewise there are so many advantages are there regarding this. And regarding this physiological and hydrodynamical factors that we maintain, one is the DO, dissolved oxygen, pH, supply of the nutrients. And okay, whenever we take this stirring process, the main problem is with the shear forces. So the shear forces are going to affect the working conditions or the living conditions of the cells. That is also going to be a problem. And in addition to that, extracellular matrix, that is also has to be taken into consideration. Next. And then whenever we look at the choice of the bioreactor, so there are mainly two varieties of the bioreactors are there. The mode of the, based on the mode of the cell cultures. Here the first one is the suspension mode. Suspension mode means the name itself indicates that the entire, there will be a slurry of the nutrient in which these cells are going to float or submerge. And the second one is adherent culture system. This adherent culture system, we can say that this is similar to that of the S system, SSF. So when those who are having the minimum bioprocess introduction, they are aware that there are two types of systems are there. One is SSF, the second one is SMF. What is SSF means? Solid state fermentation. So this adherent culture system will be similar to SSF, not the same. Let me repeat to make you understand. I am using the similarity with SSF and SMF. So if you take SSF, that is solid state fermentation, that is having the similarity with that of the adherent culture system, whereas SMF, submerged fermentation system, is nothing but the suspension type. Okay, here when we take this suspension type, that is the SMF type, we can use different reactors like stir tank, rotating wall, and wave bioreactors. Next one, please. This is a static system. This is a traditional one. Okay, this one shows. Next one, please. So this is a spinner flask where at the laboratory level we can manufacture it. So here, okay, you can go next one. No problem. So the same spinner flask is shown here, where it is having the inflow of the air and the glass jacket and untitled system. Next. Yeah, so when we take this one STBR, that is a stir tank bioreactor, it is having its own advantages. So if you look at this advantage, the main advantage is nothing but improvisation of the mass transfer. So when we take any this process industries, whether it is chemical process industry or bioprocess industry, the main things we have to take into consideration is two. One is heat transfer, the second one is a mass transfer. So they are going to play a vital role on the ultimately the final yield of the system. So in this case, improvised mass transfer is possible in the case of this STBR. And what is in this case, the control collision or aggregation or aggregate size maintenance, that is also going to be one of the important one. And here homogeneous culture environment, not only with respect to the culture, even other conditions, the nutrients, or even the pH, even the oxygen pressure, that also is possible the uniform in the case of the STBR. Next one, please. Of course, there are some limitations are there in the case of the STBR. So when we take this STBR, the main thing is higher shear stress. As we said that whenever stirring is happening, they are going to collide or bombard that particular cells. Those cells are going to be affected, their activity. That is going to be the main issue. Okay. Likewise, the many issues are there. Next one. This is a rotating wall vessel. So where continuous rotation is going to happen. Next. So this is a rotating wall vessel shown here. This is a horizontal system, whereas previous case, in the case of the STBR, we have seen that the vertical system. This is a horizontal system. Next. So when we take this rotating wall vessel, it is having its own advantages and disadvantages. If you look at this advantages, mass transfer is the good mass transfer, you can observe that one. and the next one is efficient oxidation of the medium is going to happen. But at the same time, it is associated with disadvantages. Okay, this is okay to some extent. Scale up to the production level is somewhat difficult. Next one. So this is the Wave Reactor. So what is this Wave Reactor means? This is one which is going to fluctuate it. ఒక విధంగా చెప్పాలంటే ఇది మన పార్కులో గనక పిల్లలు అటు ఇటు కూర్చొని ఒక బల్ల మీద ఒకళ్ళు పైకి వెళితే ఒకళ్ళు కిందకు వస్తుంటారు. సో ఆ సిస్టమ్ గా ఉంటుంది బయోవేవ్ రియాక్టర్ అంటే షేకింగ్ సిస్టమ్ లైక్ దట్. ఇది మన పార్కులో ఇద్దరు పిల్లలు అటు ఇటు కూర్చొని అప్ అండ్ డౌన్ అవుతుంటారు కదా. సో లైక్ దట్ దిస్ వన్. So in this case, you can find that there is a red liquid here that is present in the here. What we do means the entire what is called bio reactions are going to happen in the bags. So bags themselves are having some inlets and outlets, and then here the medium is placed in the bag, and then here what is there, simply up and down will be there, which we say even in the case of chemistry rocking. So rocking means up and down. So up and down rocking system is there. The tray is going to move from one side to the other side. So the rocking is going to help us to maintain the uniform conditions. Next one. So in this case, what is going to happen means, look at here. So this is up and downs are there. This is rocking system. So whenever the rocking motion is going to happen, the cell culture media, whatever is there, it is going to move from one place to another side in the form of a wave. That's why we call that one as a biowave reactor. So when we take this biowave reactor, it is having its own advantages. Like good, it is as we have seen. Can you go to the previous slide? Previous slide. So if you go to the previous slide, here you can observe to the bags, there are some inlet systems are there. So here the requirements, whatever are there, you can give inlet, and based on that effluent gases also can be removed. Next one, please. Next. So here, back. Yeah. So in this case, air circulation is possible because of the presence of the ports. So in this case, in this way, it is having one very good advantage. And then here, as we said that proper efficient mass transfer and mixing is going to happen, because it is not stirring, it is only rocking is going to happen. And then here, though there is a wave motion, this is a gentle wave motion. So whenever gentle motion is there, there is no possibility for the formation of the bubbles. So whenever, and similarly, the shear damage is also reduced. Likewise, many advantages are there. Next one. So okay, this bioreactor, whatever are there, this one already is there in the market, which is used up to the level of 10 liters. Bioreactors are available to manufacture neutrophils from HCS. Of course, the main disadvantage of this one is the cost of that one. So the cost associated with that one is the main disadvantage. Next one, please. Okay, next. Yeah, so this is one more, that is a perfusion bioreactor. Okay, next. So this is the working conditions of the perfusion reactor. Next time, class low, no? Okay, next. Yeah, this is the 3D perfusion bioreactor. Next. Yeah, next. Okay, this is the packed bed perfusion bioreactor. Whatever the perfusion is there, this is in the form of the packed bed. You observe this one. Here the bed is packed here. Next. This is the hollow fiber configuration. Means just like our straw, some hollow fibers will be there. So many straws like arrangement will be there, and a hollow fiber configuration. Next one. Next. Yeah, so hollow fibers, whatever I said that from the top you can observe on the right hand side. So this is just like so many straws are there. So this is hollow fibers will be present in this. There will be cartridge system. So the cartridge system is made up of polycarbonate, and then the sealant, whatever is there at the top, that is made up of the polyurethane, and then the fibers, whatever are there, and right side the cross sectional view is shown, and the entrance will be at the bottom, and exit will be at the top. Next one, please. Yeah, so this one in the industry, how they use it. Next. Yeah, this is one more, that is a fluidized bed. This is also one of the system used in the industries where air gas feed is given from the bottom. So naturally, whatever the cells, whatever are there, they will be floating. So this is there will be the whatever the cells are there, they are not going to settle down. So continuously they will be in the form of the fluid. That's why this is called fluidized bed reactor. Next one, please. So this is the experimental rig. Next. Yeah, this is about the comparison of so many types of the reactors, whatever we have seen: static flask, spinner flask, rotating wall vessel, perfused wall vessel. So how the mass transfer, flow, shear stress, strain, electrical, okay, how these are going to be vary, okay, how they are going to be, okay, whether they are going to be turbulent level or laminar level. In the case of the flow, in the case of the stress, always we require less shear stress, whether it is less or more, and then electrical simulation to what level it is there, and then imaging system is available or not. So this is about the comparison. The total information about different reactors can be seen here. Next one. And then the question arises, yes, so many are available. We have seen the parameters. How to select a proper one means, okay, the proper one we are going to select by taking some characters. What are those means? One of that one is what is the nature of the stem cell that you are going to take it. And then for what purpose you are going to use it. Okay, these are the main two parameters which are going to help us to choose a proper bioreactor. Next one, please. And this is about advantages and disadvantages. A comparison, no need to go for that. Next one, please. Next. Of course, this is okay. Till the time previously I have shown that is what is called submerged type. That is just like our SMF. This is just like SSF, solid state fermentation, like that. Not the same. It is similar, where the culture is going to adhere to the surface and then they are going to grow slowly. So in this case, if you want to go for the large scale production, so you are going to use micro carriers or three-dimensional scaffolds or micro encapsulation. These are the main three methods that we use for their production. Next one, please. Yeah, next. No need to this. Okay, because time is not enough. Okay, because already 1 hour 15 minutes is over. I want to go for the chemical system. I want to know how the softwares are going to be helpful for the chemical system. I go fastly, quickly. Yes. Please stop here. Right. So till the system, we have seen, as I said that at the beginning itself, in the pharma means don't think that only chemistry is going to play a vital role. So you must have the knowledge of the biochemical, you must have the knowledge of the microbes, then only you can work there. So that's why I have given I tried to impart the knowledge of the bio and chemical and microbial system and including the stem cells. So here in the next slides, I am going to discuss about one of the technique which is used in the industry, that is supercritical, superfluid critical chromatography, which is called simply SFC, super critical fluid chromatographic conditions. So this is the work done under my guidance. One of the student, that is Narasimha Rao, he is able to get good publications in the analytical methods of Royal Society of Chemistry. Next, he got even in the Taylor and Francis, like separation sciences and technology. Next. And then he also got in the cogent chemistry Taylor and Francis, like that. Next. This is about just what is the work done. Mainly he was able to use this superfluid critical chromatography technique for the determination of the fluorescent diastereomers. Next one. So this is one more system, that is injection formulation, that is chlorphyrabin. Next. Right. Okay. Whatever the next slide I'm going to discuss, this is the giving a little bit introduction to superfluid critical chromatography. And then how my student used the same one for the analysis I'm going to discuss. Before continuing, let me know Yeah, okay. Am I going in a proper way? Okay, my voice is clear, no issue.
Yes,
Internet
sir. Everything is clear, sir.
Sir, no problem.
okay, internet bandwidth is also no problem.
No, yes, sir. Everything is okay, sir.
Okay. Am I too faster or am I too slower?
No,
Am I
sir.
wasting your time by going slowly, or am I rushing a little bit? Can I go with the same pace or a bit slow?
Same, sir. No problem.
Yeah.
You're explaining very
Yeah.
nicely, sir.
No, sir. No, sir. Carry on.
Yeah.
You're explaining very nicely.
Yeah. So actually, I came to know that many of the participants are faculty members. So that's why I do not want to waste their time by giving the fundamentals. I gone a little bit peripheral way. Okay, and then if the students are there, if the mixed system is there, means I can go a little bit medium via way. But most of them are faculty members, so they are aware about all these systems. I do not want to waste their time. That's why.
Okay, sir.
Yeah. So right here, when we take this superfluid critical chromatography, this one is a part of the green chemistry. So when we take this one is used mainly. It is an analytical tool for the separation of the active compounds as well as the impurities. That too within less time. That is the main advantage of this one. So in this case, the mobile phase, whatever we choose, a peculiar one and a different one, that is the carbon dioxide. So why we choose the carbon dioxide? The carbon dioxide is having efficiency to dissolve the organic compounds, and then they are able to show some lipophilicity also towards some of the molecules wherever polar groups are present, oxygen containing groups like esters, ethers, etc. So that's why here we use the mobile phase. And as we know that carbon dioxide, even if it is released into the atmosphere, no problem. So that's why we can say that this one is one of the best green chemistry system. Next one, please. Of course, this same supercritical fluid conditions can be used even for the extraction. So nowadays, the extraction is going to play a vital role. Nowadays, a lot of market is there in India, mainly with respect to the phytomedicines. So from the plants, mainly not only the Ayurvedic. Don't think that I'm going to advocate about the Ayurvedic products. I'm not going to advocate the Ayurvedic products. So even if you look at the pharmaceutical products, whatever are there, they're taking the precursors. Precursors means starting molecules they are taking from the plants. Instead of preparing in the laboratory, the complex molecules, whatever are there, those precursors they are taking from the plants, and those phytochemicals they are utilizing to convert into the phytomedicines. So a lot of market is there. If you look at the entire world, the major part of the share is occupied by the Indian pharma industries. That is the importance of that one. So if you want to extract the phytochemicals, in that case, naturally, we have to go for the extraction. In that also, SFC is going to help us. Next one. No, sorry, not SFC, superfluid conditions are going to help. That is called SFE. Sorry, I said SFC. SFC means for analysis. SFE means superfluid critical extraction. Now, so here, whenever we take this SFE, that is phytochemical processing, and that one, that is having the unit operations, whatever are there involved in it, they are having a similarity is there between chemical engineering and phytochemical processing. So if you look at the mass transfer, so you can find that similarity is there, and process design, bioprocess engineering, power technology, instrumentation and measurement, environmental conditions, environmental engineering. In all these cases, those who wants to work in this phytochemical industry, they must have minimum knowledge of the chemical engineering. So ultimately, what I would like to say that whenever a chance is given to any chemistry faculty member to teach the biochemistry or related subjects, environment, most of the times we say that no, I'm a great chemist, I do not want to handle it. We should not say that, unless you have a little bit of knowledge, not only the vertical knowledge, Okay, the lateral and horizontal knowledge is also very, very important. So whenever you go to industry, that is going to help us a lot. So in the industry, if a chemist says that I have the knowledge of the chemistry, don't work on other systems, means he cannot survive for a long time. So in such cases, they must have a minimum knowledge on not only the biology, even on the biochemistry, biotechnology, and then chemical engineering is also required. So this table shows the comparison between them. So in this way, we can say that in all these unit operations, whatever are there, okay, we must have the minimum knowledge. Next one, please. Right. Now, here in the SFC, as I said, carbon dioxide is going to be used as the mobile phase. Why? Because the carbon dioxide is having a peculiarity. What is that peculiarity? It is having the advantage, it is showing the properties of the gas as well as the liquid. So carbon dioxide is showing in the critical conditions the properties of both gas as well as the liquid. So that's why they are able to extract rapidly. Why? Low viscosity is there and diffusion is also good. So out of these two, viscosity and diffusion, these are the two properties of the liquid and gas. So here low viscosity is the advantage, high diffusion is also advantage. In this way, carbon dioxide is going to be helpful for extraction. That is the main advantage. Now, when we take the density closer to liquid, therefore great extraction happens. Viscosity is closer to gas. That's why very good mass transfer is going to happen. Next one, please. Yeah, so this is the critical, supercritical conditions. So just like those who are having the knowledge, because all of us are chemists, we are having the knowledge of the what is called phase diagram. In this, just like a phase diagram, this is the critical point where you have to select that conditions to maintain. Next one, please. Yeah. So when we take this superfluid critical extraction, as I said that in the pharma industry they are going to use it. Okay, actually most of the times for the extraction we use solvents like ethanol, methanol, or water. But we say that these are the hard solvents. But carbon dioxide is considered as the soft solvent or easy solvent. Next one, please. Go back a little bit. Yeah. So if you look at this, no. Next one. Yeah. So this one, what is the advantage of this one? Means if you separate the solvent of the extraction, no need to heat much. You know that if you bring to the room conditions itself, the carbon dioxide is easily escapes out. So that downstream processing also will become easy. So when we take any industry, in addition to the upstreaming and downstreaming, don't think that these are the what is called Aryan words. Upstreaming ante నువ్వు రియాక్షన్ చేయకపోయే ముందు ఏవైతే కండిషన్ నువ్వు ఏవైతే చేస్తావో అవన్నీ అప్స్ట్రీమింగ్ అంటారు. అంటే అప్స్ట్రీమింగ్ అంటే ఏమిటి? నువ్వు తీసుకున్న రియాక్టెంట్ ని సైజ్ తగ్గించడం, అంటే పౌడర్ చేయటం, అలాంటివన్నీ ఏవైతే ఉంటాయో వాటిని అప్స్ట్రీమింగ్ అంటారు. అంటే నువ్వు రియాక్షన్ కండిషన్ ముందు చేసేవన్నీ అప్స్ట్రీమింగ్, రియాక్షన్ అయిపోయిన తర్వాత సపరేట్ చేయటము, ప్యూరిఫై చేయటము అవన్నీ డౌన్ స్ట్రీమింగ్ అంటారు. So, the upstreaming, downstreaming, processing, these are the three important wings which are very, very important. So, in that case, in the downstreaming, this the importance of this carbon dioxide can be realized. Next one, please. So, there are so many mobile phases are there. You can find that a list of the fluids. So their critical temperatures are also there. Out of all these, we can say that this carbon dioxide is the best one. Sir, next one, please, sir. Go a little bit faster. So many comes. Yeah. Carbon dioxide, NTO, and hydrocarbons like N-pentane is also used. But out of all these, nowadays adapter one is the CO2. Next one, please. Yes, this is an excellent solvent. Already we have seen that. And advantages, what is that means? Waterless, non-toxic, available, and inexpensive. So all these are the points about the CO2. Well known. Next one, please. And then here, solute solubility. You can observe that on the x-axis, if you are going to increase, automatically solubility of the solute is also increasing. So already we have seen that earlier the carbon dioxide is having or possessing the characteristics of both the gas as well as the liquid. They are going to help us to diffuse properly, and then not only that, with respect to the viscosity density also is going to help us to improve the solubility. Next one, please. Of course, effect of the pressure. Next. Yeah, so here sometimes in the case of the already we have seen about SFE, superfluid extraction, that supercritical fluid chromatography. If you take in that chromatography, sometimes we have to add organic modifiers. So these organic modifiers, just like in the case of HPLC and UPLC, this is going to help us to change the density, but not to much extent. Okay, that is going to help us. Okay, and the properties of that mobile phase is going to be modified. Okay, that is a main advantage. Next one, please. So these are some of the modifiers we use in the case of SFC, like methanol, ethanol, propanol, or acetonitrile. Next. And then the stationary phase, whatever is there, just like in the case of others, even here you can use either packed or open tubular. Most of the times in the case of analysis, we prefer the latter one, that is the packed one. In the case of the extraction, the better one will be open tubular one. That is the main difference. Next one, please. So as usual, the packing material will be siloxanes as usual. And then here the detector system will be there. So just like in the case of SFC, even here in the case of GC, mainly we prefer FID, that is a flame ionization detector, because what is the reason means as it is involved with the gas. Next one. So here this is about the carbon dioxide. We have seen so many advantages. Okay, the main disadvantage is nothing but heavy investment. What is the investment means? Okay, if you want to store those gases, okay, naturally you require a very good, just like your LPG, liquid petroleum gas. Man intlo ganaka ee vidhanga cylinders untayo, like that storage conditions are required. Transportation is also difficult. Such disadvantages are there. But regarding the conditions like mildly critical conditions, non-combustible nature, non-corrosive, low price, poisonous less, harmless, all these are the advantages. Next one, please. Next. Yeah. So this is the flow chart for the supercritical carbon dioxide extraction. So when we take this one, just look at here. So here at the top, there is presence of the carbon dioxide cylinder. And then after that, carbon dioxide from the already carbon dioxide will be present in the high pressure. But further, if you want to improve the pressure, we use the compression, and proper maintenance is possible with the help of the controller. And then the carbon dioxide, which is turned into supercritical fluid conditions, then it is allowed into the system. And then here you are using here one more extractor, that is tributyl phosphate. It is also compressed and then introduced into the system. Okay, not only in the case of the phytochemicals, this one the fluid critical chromatography is also used in the case of the nuclear processing industry. As I said that I am discussing about the processing industry, maybe pharmaceutical, or it may be phytochemical, or it may be nuclear, what it may be. So in the nuclear processing industry, so if you want to enrich the uranium, so in the enriching of the uranium, this is going to be helpful a lot. So here we take the uranium solution, and then this SFC carbon dioxide along with the TBP, tributyl phosphate, is permitted here, and then there will be a trap, and then followed by carbon dioxide release will be there, where if you reductive reducing valve means simply if you open it, so carbon dioxide escape out. That is the easy way of vent. So in this way, in the nuclear processing industry also, this supercritical fluids are used. Next one, please. Yeah, when we take this superfluid critical extraction, there are two types are there: liquid SF, solid SF. What is liquid SF? Liquid SF means, for example, if you want to remove or reduce the alcohol from the beer, you know that nowadays the beer consumption is increasing day by day, is consumed by all types of genders. So some of the times they may require less kick. Okay, so they want to reduce the existing alcohol percentage. That removal is possible with the help of the SFE. Okay, gents, they prefer to enrich the alcohol. Ladies prefer to reduce the alcohol content. For ladies' purpose, this SFE is going to be helpful. Okay, and then in the case of the solid SFO extraction, so this is called leaching. You know, the leaching is nothing but from the solid mixer. If you are going to extract one of the what's called solute, we call that one as leaching. For example, I want to give a simple example so that you can understand it. If a mixture of sand and sugar is given to you, if you are asked to separate them, hand picking takes a lot of time. How to separate it? You add water, shake it. So sugar dissolves in water, you filter it, you get the sugar water, evaporate, you get the sugar. So in this case, what happened? The sugar is dissolved, but not the sand. So preferential solubility is used in the case of the leaching. In a similar way, we are also using this SFE to remove the caffeine from the coffee beans. So coffee beans nunchi caffeine removal, that one is going to be useful. And then mainly last one, rose water. So once upon a time, the rose water used to prepare with the help of the what's called by sending the steam into it. You know that extraction. So they take the rose petals into which the water is heated and converted into steam. The steam is permitted into the vessel containing the rose petals, and rose petals fragrance is taken out by the steam, and then steam is condensed, and then you get the water, and then that is called rose water. So those who have visited this what is called Rajasthan, by default Jaipur, Udaipur, that area, okay, whenever you board any bus, they take to that industry. Probably all of you have seen that. So, but in that case, there is some disadvantage. Sometimes the fragrance containing compounds are very fragile. Fragile means easily break down. So in such case, SFE is going to be a very good, helpful one. So from the petals of the rose, this flavor can be extracted. Next one, please. Yeah, so this SFE is having natural products. Okay, natural products means nothing but a part of the pharma industry. Okay, in the drugs, foods, pesticides, surfactants, like that, so many applications are there. Next one, please. Yeah, in the natural products extraction. So if you take the if you want to separate, or if you want to learn how much cholesterol is present in the fish oil, so from the fish oil, cholesterol estimation can be done with the help of this SFC. Next one, please. Yeah, caffeine extraction, hops, and nicotine. Hops means this is nothing but okay, this is a type of flowers. From those flowers, the color can be extracted easily. That is the main advantage. Okay, next one. Yeah, this is about the large scale SFC extraction. Okay, in the case of natural products, in the case, okay, we can use it. Next one, please. Yeah, so this is about the flow sheet regarding the enrichment of the ethanol using supercritical carbon dioxide. So what we do means, this is the vessel, or actually industry they call that one as the reactor. Of course, as we are doing the extraction, this is also called extractor. So this is the extractor. In this extractor, what we do means we are giving the feed. What is the feed? Feed is nothing but the ethanol plus water mixture. Okay, from the bottom, you are giving the mobile phase. What is the mobile phase here? Nothing but the carbon dioxide. So the carbon dioxide is the inlet. Look at here, right hand side carbon dioxide that is compressed and it is fed at the bottom. And what happens? The ethanol feed, water mixer feed, and this gas, they will be in the countercurrent. Countercurrent at the moment, what is the meaning? Ethanol will be moving down. Ethanol plus water mixture is moving down and carbon dioxide is moving up. So countercurrent direction. So countercurrent, co-current, out of the co-current and countercurrent, always countercurrent extraction is the best one. And then after the removal, whatever the leftover at the bottom, unnecessary thing is we call raffinate. And then the extracted one is separated and carbon dioxide is recycled. This is a simple flow sheet where the separation of the ethanol from the ethanol water mixture can be done. Next one, please. Yeah, this is about coffee beans. So instead of the alcohol water mixture, you are giving coffee beans from the top, and then compressed carbon dioxide you give, same is going to continue. So the extraction of the caffeine extraction from the coffee bean is going to happen. So coffee beans are given from the top, pulverized. Pulverized means broken down, powdered one. Pulverized coffee beans are given from the top. From the bottom, the carbon dioxide of the compressed gas is given. They go in the counter current, that is opposite directions. The carbon dioxide, whenever it reaches at the top, so it is going to extract the caffeine, and then pressure is reduced, and then at the separation, here caffeine is collected at the bottom of the separator. The carbon dioxide, whatever is there, once again compressed and recycled. Raffinate means after the extraction of the caffeine from the coffee beans, pippy edaithe untundo, leftover material is called raffinate, that is collected at the bottom. Next one, please. So look at here, this is a little bit animation, how superfluid critical chromatography is going to work. Just look at it. Yeah, the left hand, let's start from the left hand side. The first one is the mobile phase carbon dioxide containing cylinder, and second one is the compressor, nothing but the pump. Compressor means which is going to increase the pressure, and then this is the coil. This is the column in the form of the coil, and the third one, this one is the injection system, where the sample is injected here, and the entire arrangement is box represents the oven, where the proper maintenance of temperature in the thermostatic conditions is possible. After passing the sample and the mobile phase in this column, fifth one, in the form of the coils, they are well separated, and here the sixth one is the detector, and the seventh one is the chromatogram. So here we get the concerned chromatogram here. Right, next one please. Here the vapors whatever are moving up, those are nothing but vapors of the carbon dioxide. Of course, these are the different types of the injectors, like loop injectors, inline injectors, in column injection. Different types of injectors are there. Time is not permitting me. Let me rush a little bit. Next one please. Yeah, next, of course, the same thing. Next. So this is about the publications done by my student. Next one please. Yeah, okay, this is one of the molecule taken by my student, that is the fulvestrant, and this is mainly used for the breast cancer, and then it is having the 6 asymmetric carbon systems, having some stereo system, as it is having that one 2 diastereomers are present. Those diastereomers, whatever are there, called A and B, we have to separate them. The separation is very much required. Of course, in the USP, that is the United States Pharmacopoeia, already methods were given, but it takes 30 minutes. But we have to reduce that one. For that, my student has chosen the system. Next one, please. Yeah, this is the chromatograms obtained from the current study. And next one, if you look at this one, at the bottom, as per the USP. So if you look at that one, well separation is there. The top one is done by my student. The bottom one, same adapted whatever given by United States Pharmacopoeia. So compare it to the USP procedure, whatever the procedure developed by my student is the best one, where the resolution is also very high. Next one, please. And not only, sorry, previous one. Look at the time. Okay, USP requires 30 minutes, whereas that system developed by my student requires 2.5 minutes or 2.6 minutes. Not only that, resolution, how they are well separated. Look at the top one, my student done the top one, they are well separated. Whereas look at the bottom one. So the bottom one, whatever is there, still their resolution is not much. So compared to the USP, my student did well. Next one, please. Yeah, columns. So they have selected. There is some systematic procedure is there. Time is not permitting me. Move. Next. Yeah. So like this. Next. Yeah, so as usual, he validated as per ICH guideline Q2R1 system. Next one, please. Next, this is one more. Yeah. Next. Yeah. Okay. In this case, also almost 10 times the runtime is reduced. That is the main advantage of this one. Next one. Yeah. So, okay, as we said that this is the green system. Carbon dioxide is present in it. Next one. Here with so many impurities. Okay, he has taken almost 8 to 9 impurities. They were well separated. Next. Yeah, so stop here. This is the peak purity plot. Already you are aware that is just by comparing the purity angle with that of the threshold, we can say that whether the peak is in the homogeneous or non-homogeneous. In this case, purity angle is less than purity threshold. That's why we are able to say that peak is homogeneous. Next one, please. Next. Yeah, so okay, different okay condition parameters were tested by my students, which are under ICH guidelines. Next, yeah, force degradation studies were also done. Next, yeah, this is about methods, whatever are there, control conditions, whatever chosen by him, or even robustness is up to the mark, no issue. Next, next. Right. Okay, so okay, let me know. Can you bear with me a few more minutes so that I can discuss about what is called
No problem, sir. No problem.
DBE method?
Okay, sir.
Just 1 second, 1 second.
Okay, sir.
Right. Just 1 second. My system is a little bit slow. Just 1 second. Madam, Jyoti Madam.
Sir?
Madam, I am sending one more PPT, madam. A few slides were missing in the previous one, whatever I sent.
Okay, sir.
I am sending. Just please share that one.
Okay, sir. Okay, okay.
Madam, your email ready? Madam? Hello, Jyoti madam.
Sir, JMJ Chemistry.
J.
JMJ.
1 minute, sir, 1 minute. JMJ
Chemistry.
chemistry.
2022.
What's that?
2022.
JMJ chemistry 2022.
Oh, yes, sir.
JMJ.
Sir, a chat box to type just student, sir.
Yes, sir. I sent it, sir. Just check once.
Okay.
Oh, okay, sir. Wait.
Whether you received it, let me know.
Yes, sir.
No, no,
Received,
no.
sir.
Oh, my class.
రాలేదు మేడం చూస్తాను.
It takes just 1 second, man, because mine is Yahoo. Yahoo always
Okay.
takes a little time.
ఇంకా రాలేదు మేడం ఒక్క నిమిషం.
MTech.
Sir?
Got it, madam?
No, sir. No, sir.
Yeah, I am trying to send it again.
Okay, sir.
మీరు ఎక్కడ చూడమాట
We started Untitled, right, sir? In 2019, we started Untitled. Before of that.
ఒకసారి అజయ్ గారు అన్నమాట.
Okay, sir. Okay.
Got it, madam?
Yes, sir. Yes.
Got it. Okay, okay.
which slide, sir? There are 246 slides are there. From where can I start, sir?
No. You first share the screen so that I can say.
Okay, sir. Okay.
There will be some design of experiments will be there.
Okay, sure.
Yeah, go, sir. Go fast, a little bit faster. Yeah. Previous slide. Yeah. So, just one second. Sorry, I was unable to share the real one, and that's why it's 10 minutes wasted your time. Sorry. Okay. Please look at that slide. If you look at the slide, when we take the research, there will be a sequence of steps will be there. In that, at present, I am concerned with the second stage, that is the development. So when we take this development, always the development has to be done, and then development of the here analytical development means. So as per the ICH guidelines. Next one, please. So here we are going to use. I'm going to discuss about DOE. That is design of experiments in analytics. Of course, the same DOE can be extended even in the case of synthesis. As I said that nowadays software help is going to help us to finalize the things at an early. So I will discuss. Madam, I will try to finish within 10 minutes, madam. Don't worry.
No problem, sir. Carry on.
Of course, understandable. No worry for you and me, for participants. I will try to finish. Okay. Next one, please. So, okay, actually this DOE approach followed by one of my students, Nogendra Prasad, who was able to get good publications in a Journal of Liquid Chromatography and Delayed Technologies of Taylor and Francis. Next. From Oxford publishers JCS, the Journal of Chromatography Sciences. Next. The same in the Taylor and Francis journals. Next. So this is the one. Okay, with this my students' research, I want to give how it works. I'm not going to discuss much about my research. I say just what is what. So actually, this DOE approach was given very long back in 1920s. Mainly, it was applied for the agriculture. Now here, if you want to do the experimentation, if you want to reach your target or goal, there are some approaches. So what are those approaches? At present, we are following trial and error method. Trial and error method means we try for it. Okay, whether working or not, we go for the next parameter like that. Trial and error, which is not at all acceptable. Nowadays, many members are following one factor at a time. So one factor at a time means, let us say 5 parameters are there. Out of the 5 parameters, 4 parameters they keep constant, and the 5th parameter they change. Next time, 5th parameter is kept constant, 4th parameter is changed. It means out of 5, 1 parameter they keep constant and change the other parameters. They keep 4 parameters constant and change only 1 parameter. But that is also not good, because optimum conditions you cannot get. So what is the best method means? Better to adopt DOE. So what is the importance of the DOE means? At present in the analytical wing we are using. In future, as per the FDA, not now, in future it is going to happen. FDA is going to enforce the usage of the DOE for the purpose of the what's called experimentation. So what is going to happen in this case? In this case, a systematic multi-factor experimentation is going to happen, and then the performance will be very robust, and then stability of the process product also increases. That is the main advantage. So robustness, reliability, everything is good. That's why this DOE approach is more and more adopted. Next one, please. Yeah, look at here. At present, let me take only 2 parameters. No, for the process, I am going to take only 2 parameters. I am going to take a synthesis. In the synthesis, I am taking 2 parameters. What are those 2 parameters? X-axis, temperature. Y-axis, pH. So both of them are influencing my system. In order to get the final product yield, target yield, my target yield is more than 98%. So the goal, whatever is there, that is why more than 98% will be there. Whenever you look at here, when the temperature between 60 to 90, and when the pH is in between 4 point something to 6 point something. But here, what is going, if you are deviating from the temperature, for example, you are increasing the temperature. Generally, there is a myth. If you increase the more temperature, if you get the product very quickly and more product you get. Temperature పెంచితే బాగా product వస్తుందని మన అందరికి myth ఉంటుంది. అలా కాకుండా if you increase the temperature beyond 90, what is happening here? Yield is decreasing. You are getting only 94%. So in this way, what is going to happen means here, based on the requirement of your goal, you have to properly maintain the pH and temperature. For example, if you maintain temperature less than 60, you are able to get only 90. If you maintaining more temperature more than 90 degrees centigrade, you are reducing the yield. So from this, what we can say that proper maintenance of the temperature, proper maintenance of the pH are required. So how to know that? I am able to write some lines here, some circles. Such circles we can get from the DOE. That's all simple. Understood? So here we get the circles. So from the circles, we can know what are the optimized conditions. From those conditions, optimized conditions, from that circle, you can choose it. I'm not going to discuss about much terminology. I'm giving simple layman language, that's all. Next one, please. Yeah, so why means, as I said that, here, always whenever you are going to develop any method, first screening happens. So screening means number of the experiments will be there. So the screening can have minimum number of the experiments. That is the main advantage of that one. And then whenever some variables or factors are there, how they are going to affect each other. That is also very important. Process optimization is required. And you want to develop the time. So instead of doing many experiments, you can reduce the number of the experiments. When the number of the experiments are reduced, automatically development time is also reduced. And process, whatever is there, sometimes you are forced to vary the process parameters, conditions. Even then, it is going to be reliable or not, we come to know. So likewise, all these are helpful with the help of the DOE. Next one, please. Next. Yeah, so in the case of DOE, whenever we take this DOE in the pharma industry, for analytical R&D we can use, formulation R&D we can use, for the API synthesis also we can use. I am going to show the first category, analytical R&D I am going to use. Of course, you can also extend even to formulation R&D and API R&D also, that's a different story. Next one. So here what happens? A series of the tests we run, and then here we ultimately what we are going to go means look at the second paragraph. Second paragraph red marked. The relationship between factors influencing the process or affecting the process and output of the process we have to relate. That is relating is done with the help of the DOE. In this DOE, what we do, initially we do some experiments based on the factors which are influencing. We test with the help of the ANOVA. ANOVA means already you know that analysis of the variance, and this is the one of the analytical tool in the statistics, so that we come to know about systematic factors and the random factors, and then we take the help of the regression analysis. Next one, please. Yeah, so as I said that I am going to optimize the analytical R&D conditions. I am taking the chromatography conditions. Left hand side input to factors, right hand side output to response. Input factors left hand side, output is there on the right hand side. Input always x-axis variables, output always y-axis variables. So here whatever the input variables we call here as the control variables. These are the pH of the mobile phase, column temperature, solvent ratio, flow rate, buffer concentration, column chemistry. All these are the control variables. What are the response variables? Resolution. Resolution, how much we require? NLT2 means not less than 2. Tailing factor, not more than 2. Runtime, always less than 1 minute. So like that, some output responses are fixed. Input factors, whatever are there. So how can we optimize the process? That is done with the help of the DOE. Next one, please. So when we take this DOE, we can say that mainly two parts are there. One is factorial design. The second one is RSM, that is response surface methods. What is factorial design? The design happens. In this case, different types of designs are like full factorial design, fractional factorial design, placket Berman design, like that. and RSM, response surface methods, how we are going to represent in the form of the graphs or sheets, whatever are there. There is CCD, Central Composite Design, Box Benham Design are there. Nowadays, whatever the software we use, that software is a mixture of the above two. There is Factorial Design as well as RSM. Both are mixed, and then they are available in the market in the form of design expert. That is also called StatEase or Mini-Top software. These are the two softwares which are coming under the category of the mixed design technique. Those are Factorial Design as well as RSM. Next one is. Yeah, so in this Factorial Design, we are going to take level and factor. So we are going to take factor will be here. For example, to make you understand, let us say that level is 2, factorial is k, then 2 to the power k you have. So factor always power to the, okay, that is the full factor. Fraction factor means that one is half factorial means 2 to the power k minus 1. And then in the case of 2k to the power k minus 2 means there is 1/4 of the factorial. So likewise, next one. So this is about the factorial designs. This is a level 2, or 2 to the power 1, or 2 to the power 2, or 2 to the power 3. Number of the runs will be done. Look at here, first one, 2 to the power 2 means, first case only 2 runs are done. 2 to the power 2 means 4 runs are done. 2 to the power 3, 8 runs are there. 1 to 8 runs. Are you able to see here? Left hand side, right hand side, 3 tables are there. The table consists the number of the runs. In this number of the runs, the factors whatever are there, plus or minus 1 conditions are shown. Next one, please. So likewise, you can go up to 2 to the power n. Here from 2 to the power 1 to 2 to the power 2 to 2 to the power 9. These are given. Number of the factors, how they are going to show. Next one, please. Yeah, so response surface methodology will be there. This one, in this case, what happened means, so optimized settings of the significant figures is going to happen. As I said, 2 popular RSM designs are there, CCD and Box Benham design. Okay, next one, please. Yeah, so this is the response surface methodology. In the case of CCD, you can observe that first one is 2-level factorial design. And the actual points are there. So, okay, you can observe that horizontally or vertically, they are out of the box lines. Next, you can find that central means it will be exactly on the lines. So, the bottom one is the CCD3 factor. Next one. Yeah, are you able to see here? So, here you can find that exactly these stars, whatever are there, they're exactly on the lines. That's why we call them as face centered. There's a difference. So face centered either 2-factor or 3-factor. Next one. Yeah. So this is different from the previous one. So this is a Box Benham design where 2 factors and 3 factors you can find that. In this case, okay, what you can observe that no corner points are there. Only central points are there. There's a difference. Next. Yeah, so when we take, there is some sequence of steps are there. First, we have to define the problem and objective. We must be able to identify the control variables. Then we have to define a response variable. Then we have to choose the experimental design. And then we have to execute the experiment. Then we have to carry out statistical multivariate analysis. And then we are able to get the design space. From the design space, the optimizing conditions can be obtained. Okay, and then we do the confirmation analysis regarding the edge failure analysis or final recommendations. Next one. So in this case, pH scouting, my student has taken that condition. First, pH is one of the parameter. pH scouting studies were done, but not factorial design. Okay, if you look at here, pH is 2 and they have gone up to 6. The pH 2 is the best one. So based on the peak retention times and system suitability parameters, it was decided both for the sample as well as the impurities from impurity A to impurity E. So the tailing factor and theoretical factors are within the limits. So the theoretical factors minimum number here is a very good number, pH2 more than 3,500. Tailing factor whatever is there less than 1.5. Next one. Yeah, here initially poor retention resolution is there. After improving, how it will be there. Next one. Yeah, so these are the mobile phase composition and solvent temperature. These are the two variables they have taken, response variables, and they have changed. Next one. Yeah, so experiment design is done with two number because already I have said that temperature and other parameter, two parameters, and then response variables are given here. Next one. Next.
Wait, sir. Okay.
Yeah, so this is about design of experiments and obtained results. So when we do the preliminary experiments, we give to the system. The system has given 8 experiments. Based on those 8 experiments, 8 experiments were performed. Next one. And then we do the DOE. First, we do the ANOVA parameters verification, where we are going to take the quadratic model, and the p-value always expected less than 0.301, and if it is 0.05, then we can say that the selected model is significant, and the difference between the R values of the adjusted and the predicted model is more than 0.02, then we won't accept. It must be less than 0.02. Next one, please. Yeah. So in this case, the adequate precision is there. That the ratio, whatever is there, that is more than 4. That actually we require generally 2, around 2 to 3. Here more than 4 is there. Next. Yeah, so this is about the perturbation. Perturbation means simply we are going to disturb the conditions. Okay, if you look at on the y-axis, resolution is taken, and whereas on the x-axis, deviations are taken, and it is showing how the perturbation is happening. Next. Next. Yeah, this is about 3D plots where the effect of the input factors on the resolution we can understand by taking the two parameters, column resolution and mobile phase on the 2 axis, where the third axis, the resolution was taken. Next. And then this is DOE observations. Okay, whether the resolution or USP tailing significant or not, we can say. Next. Yeah, so this is about DOE overlay plot. Wherever yellow color is there, in that yellow color you can choose the any one point. My student has chosen a point with a flag, where you can observe that the red mark, the red point shows exactly at middle the chosen point. Next. Yeah, so in this case the resolution is very high, more than 3.7. So well separation can be observed. So in this way, well separation can be obtained with the help of the DB. Thank you. Yeah, you can stop sharing. Yeah, that's about from my side. Sorry, I have taken I think 10 minutes more. Actually 2 hours was given to me. I have taken 10 to 12 minutes more.
Any queries on organ, madam?
Any queries? Please ask to sir
Please.
to clarify the doubts.
Please.
Any participants? Sir is ready to give the answers. Write in chat box also. Sir is ready to give the answers.
Good morning, sir.
Please, madam.
I'm Dr. Sarojini,
Madam,
JMJ College, Zoology Department, sir.
please, madam.
Mine is not a question, but I want to appreciate you and your scholars.
Yeah, madam.
You are making your students, your scholars to do very great work, sir. Stem cell technology means now it is an advanced one, which is very much needed for the present situations in the present scenario in the
Yeah, ma'am.
regeneration medicine. So I appreciate your scholars and your work and your able guidelines regarding the drugs and all these things. Really, it's a great work. Thank you, sir.
Okay,
It is
thanks to my
giving
scholars. Thanks
it
to my
is
scholars who strengthened
uh,
my CV.
yes, it is giving the immense pleasure to me. Stem cells are really reading regarding stem cells and their work in our body. Really, it is a miraculous thing which was given by God, and the work whatever you are doing, you people are doing for the beneficial and for this society. It's a great job. So I thank you, and also I congratulate to you and your scholars. Please convey our wishes and our blessings to your scholars also.
Okay, ma'am. Thank you.
Thank you, sir. Thank you.
Anyone? I think nobody is there, sir. Thank you, sir, for your outstanding presentation and sharing your rich knowledge. We extend our immense gratitude to you for your availability to the resource person of the day. Your session was very informative, inspiring, and excellent. That made this day very enriching and helpful. So once again, I thank you all for making this very effective as well. Now I hand over to P. Kirtana to propose vote of thanks.
It's my privilege to propose to vote of thanks. I would like to thank today's resource person, Professor Ratnagaram Venkatnath, for his excellent and outstanding presentation. Sir, you enlightened us clearly about types of innovation, contribution of GDP in India, how this digitalization is going, transforms the entire society, different ways of pharmaceutical companies are going to using data to drive innovation and value, ultrasonics and its medical applications, SFE types, extractions, and DOE in analysis. I convey my deep regards and heartful thanks to our correspondent, Dr. Sister Teresa Magade, for her support. My sincere thanks to Dr. Sister Shaini KP, Principal of our college, for her help and support. And special thanks to convener of this international webinar, Dr. K. Nirmal Jyoti, HOD of Chemistry. Thanks to all. We are binding up today. Will you see you tomorrow's session at 10 a.m.
Thank you, sir, for your wonderful presentation on your
Okay.
topic, sir.
Thank
Thank you,
you.
sir. Thank you once again, sir.
Thank you. Thank you very much. Okay, bye, ma'am.
Bye, sir.
Bye, ma'am.
I'm closing this.